Greetings everyone and welcome to my channel. In this video we are going to talk about Mitch package. Mitch employs a statistical method known as rank MANOVA to identify groups of genes that show enrichment in multidimensional space when compared to the background. Because RNA sequencing data is prone to bias, a normalization step is essential. In our case, the DSEQ2 package on our software was selected to do the normalization step. Then we ran the results functions in order to extract the results tables from a DSEQ analysis giving base means across samples, log-to-fold changes, standard errors, test statistics, p-values, and adjusted p-values. The results tables will be the input data to run our Mitch analysis. Now let's get started. Firstly, we install the Mitch package with bioinformatics with the first command. Then we upload the following libraries, which are Mitch and the quantitative set analysis for gene expression library. We upload the libraries. Then we upload our data with read.delim function, which reads the tab delimited files. First file and the second file. Then we download the GMT file. The GMT files are a commonly used format for list of genes used in pathway enrichment analysis. GMT files can be obtained from the Actome or MSEQ database. In the description section of the video, I put the download link to download the GMT file, which contains the gene symbols. With the read.gmt function, we can read the GMT file. Now we upload the GMT file also. Before performing Mitch analysis, we must remove the duplicated gene symbols and the NA values inside the data. To remove the duplicated gene symbols, we use the duplicated function. Exclamation mark to remove duplicated gene symbols. Now we remove the NA value by using NA.omit function. Let's run that code. Now our data is ready to perform Mitch analysis. Firstly, we create a list that is made up of two variables, normal cancer 1 and normal cancer 2, both of which contain gene expression data. X, for example, equal to list cancer 1 equal to our data normal underscore cancer 1, and cancer 2 equal to our data normal cancer 2. Then this list is used as input data for the Mitch import function from the Mitch package, which calculates a summarized differential expression metric by multiplying the sign of the log fold change by the minus log 10 of the p-value. If this behavior is not desired, Mitch import can be bypassed in favor of another scoring metric. Then we specify that the analysis conducted using DSEQ2, the gene IDs are located in the gene symbol column of the input data. Y equal to Mitch underscore import, then X, our list of data, DE type equal to DSEQ2, because we perform normalization with DSEQ2, ID column equal to the name of the gene IDs in our data. In our data, it's called gene underscore symbol. So I will write gene underscore symbol. Let's run that code. Then we calculate the degree of enrichment by using Mitch calc function. There are 3 different conditions can be used by the end users to prioritize the visualizations of the results: statistical significance, which reveals the gene sets with low p-values, or effect magnitude, which reveals gene sets with large S or enrichment scores, and the standard deviation, which reveals gene sets with inconsistent regulation. We put the imported data, which is Y, then the gene sets, which is the hallmark symbol variable, and priority, either significance, effect size, or standard deviation. And lastly, in the code, we have to put the minimum number of genes required in a set for it to be included in the statistical analysis. The default is 10. Let's write our code regarding significance. Write get underscore calc, put Y, works, symbols, quality equal to, write significance, then minimum set size equal Y, for example. Regarding fixed size, write calc Y, hallmark symbols, priority equal to effect, the minimum set size, like lastly, standard deviation equal to calc by what works, symbols, priority equal to standard deviation, min set size equal Y. Let's run that code. In order to produce HTML reports, we have to use Mitch report function. So for significance report, Mitch underscore report function, then we'll write significance and move our report, and then put the name, HTML. Let's run that code. And regarding the Mitch report for effect size and standard deviation, we use the same code. Example effect report and then effect size and the name of the report, mathematics effect on .html. And for standard deviation or word score report standard deviation Then we run the code in order to obtain the effect size and the standard deviation reports also. Now we obtain 3 different Mitch reports for significance, effect size, and standard deviation. Thanks for watching. I hope you enjoyed the video. If yes, then please don't forget to subscribe to our channel, hit the like button, share the video, and leave your comments under the comment section. Until next time, happy analyzing.